It's a pleasure to welcome Professor John Nagle from Nagle University, Pittsburgh, United States here today. He started out as a theoretician really, and got his PhD at Yale with Lars Onsager as advisor, and did his first postdoc with Michael Fisher. And then after a while he realized that experiments is a too serious business to leave to the experimentalists alone, so he started doing experiments also in biological physics. And today we will hear about biological membranes, how you do X-ray scattering on something that is really not crystals, but more like liquid crystals. Thank you.
Thank you, Ollie. I got to remember to turn this thing on. Does that sound good?
Yes.
Okay. So thank you, Ollie, for the kind invitation to come and visit. I'm very impressed with KTH, and it gives me an opportunity to continue interacting with Ollie, and I'll tell you about some of that later on. Just an amendment on Ollie's introduction. Perhaps one of the reasons I'm doing experiments now more than theory is that as you get older, the brain cells start to die off, and so doing theory was getting too hard. Besides, in biological physics, things are a little bit messy, and often doing ab initio theory becomes harder, and so and we needed the experiments. So you've seen the title, which I guess was announced earlier, and the general area is one of biomembranes, and if you don't already know it, every cell has a membrane. In fact, most cells have more than one membrane, and it's a very functional part of biology. I've given you here the physicist's view of the cell. Of course, it's a spherical cow, but even if you draw the spherical cow, you've already got a membrane. So I won't go too much into the biology of that, but it's a challenge for experiment and for theory to deal with the structure of biomembranes. And of course, biophysics, we really are interested in the function, but biophysics is predicated on the assumption that you need to know something about the structure in order to really understand the function. And I'm focusing on the structural aspects of things. Also, another thing up here is, and I'd like to keep on emphasizing this point, is that biological physics, okay, so here I'm saying my biological physics is inspired by biomembranes. I would say that in general, the definition of biological physics is physics inspired by biology. This is actually a Hans Frauenfelder comment. And I'm going to try to convince you that it's not just applied physics, although there's nothing wrong with applied physics, of course, but that it really is physics, and I'd like to emphasize that later in my talk. So the next point here is, technique-wise, my main focus right now is on X-ray scattering, but it's not crystallography. If you're in biology, you think of X-ray scattering as protein crystallography. Very big, very important field, but this is not crystallography. As a physicist, I say not boring crystallography, because my prejudice is that a lot of the crystallography is pretty routine. But the reason it's not crystallography is that the samples that we use have order, but they don't have complete order, and they're liquid crystals. And so we're in the field of physics of liquid crystals here. And my major takeaway message, not just for physicists, but also for biophysicists, is that diffuse X-ray scattering, which I'm going to focus on, provides more and better information about membranes than traditional diffraction methods. And later I'll explain what I mean by traditional diffraction methods in this context. Now, I have a specific topic that's fairly recent with us that I would like to use as an illustration, and it has to do with the effect of cholesterol on lipid bilayers, and this is something that I've interacted with Ollie on in the past, and I'll tell you a little bit about that later on. But the paper that's published is a PhysRev letter from last year, and I want to follow up on that a little bit in this talk. Okay, so now the first thing is that biology, whether we as physicists like it or not, is based on molecules. And we have to know a little bit about our molecules in order to be able to relate to what we're studying. So this is one lipid molecule, and there's the usual chemist structure here. And one of the things that I like to do for physics departments is to teach them how to pronounce the names of lipid molecules. And so here's this name. You can all read it. And I'm going to teach you how to pronounce it. I've shown you a little bit of a clue as to how to pronounce it by highlighting certain things in red. And the way you pronounce it is DMPC. That's a lot easier than dimestryl cis-glycerol phosphatidylcholine. Okay? So now that you're beyond the hurdle of the terminology, DMPC, let me emphasize what that molecule does and why it's interesting physically. The molecule has different pieces, and the piece up here is traditionally called the head group. It has charges on it, positive, negative charges. It's a dipole, but whenever you have charges, you can lower the energy of those charges by putting them in a high dielectric constant medium. And of course, in biology, the high dielectric constant medium is water. And so these head groups like to go into the water. They're called hydrophilic, this name down here. The next piece of the molecule is this backbone. I'm not going to get into a lot of details about that backbone. It ties together the head group and the tails, or the hydrophobic chains or tails, and there are two of them on typical biological molecules. And they're hydrophobic because water doesn't like them. Oil and water don't mix, and so that's why they're called hydrophobic. Now, let's see. The next point is, those of you who don't do organic chemistry, let me just emphasize, because I need this for later on, what this symbolism means. So I have these things here. I call them chains or tails, but I haven't told you what these are. And what they are is shown here. They're hydrocarbons. CCC is these three bonds here. And they have hydrogen sticking off to the side. These are called saturated hydrocarbon chains because they have as much hydrogen as you can get on them. They're the things you're not supposed to eat, of course, but lots of lipids have saturated hydrocarbon chains, saturated fats. Okay, next slide, and I realized at the last minute that I really should have asked Ollie for one of his pictures instead of a picture from another. So up here in fine print, I have another one of my collaborators who actually gave us this particular picture. And the point of this picture is that ball and that sort of stick model that you saw on the last slide had things very regularly laid out in a schematic diagram. But when you actually put these things in water, and you do it in a simulation, what you see is that those regular hydrocarbon chains that I showed on the last slide are not regular at all. They come up here, they do a twist, they do a turn. Here's the end of them. So the color code down here is that these are the ends, the so-called terminal methyl groups. These are the CH2 groups that I emphasized on the last slide. And then there are all these things in the backbone. The oxygens are red. The glycerol is brown. The phosphate group, which is very important, is the green part. Choline is near the end. And the water is kind of minimized here, but there's a lot of water in this simulation. It's the sort of the blue murky background. And of course, this is nanoscience. For those of you who like applied physics, this is nanoscience, which biology discovered a long time ago. These are very thin, 4 nanometers thick, roughly. Okay, now, I have to come back because for this part of this talk, I need to tell you more about lipid molecules to make an interesting comparison. I've already told you about this one up here, it has the name DMPC, but that's just an acronym. And I'm trying to emphasize here what it is that distinguishes that from the other molecules that I want to show you. And the distinguishing feature is that if you count from here to here, you have 14 carbons, and all of them are saturated. This wasn't the most dramatic, this was not supposed to be the most dramatic part of the talk. I have someplace else, this should have waited a little longer. And I'll explain the zero in a moment. So there are two of these chains that are 14 carbons long. Here's one, different acronym, but the important feature is it's a little longer. Has a couple more carbons on each chain. And here's the saturated motif again. But now there's another motif on these hydrocarbon chains. And this one is unsaturated double carbon bonds, where there's a double bond here, and it doesn't have as much hydrogen. That's why it's called unsaturated. It could have more hydrogens if you added them. But this is an unsaturated double bond. And here's a molecule with an unsaturated double bond. It's on this chain right here. And this one's called POPC, but again, the best way to think about it is it's 16 zeros, so there's 16 carbons on this one. And now the zero means no unsaturated double bonds. This one has 18, and it's 18-1 with one unsaturated double bond. The difference between unsaturated and saturated is the thing I want to focus on in this talk. Here's one up here where each chain has one unsaturated double bond. And here's one here which is longer, but it also has one unsaturated double bond on each lipid. I didn't draw a box around them here. Now, the other key player in this talk in this paper that we just did is cholesterol. And cholesterol is a lot different than the other lipids that I've shown. All these other lipids have the same head group. There are lipids with different head groups, but I'm just focusing on these so-called PC head groups, phosphatidylcholine head groups. But cholesterol is quite different. This might be a little bit hydrophilic, but mostly it's a rigid ring structure which doesn't have the flexibility of those lipids that I showed on the MD picture. They're much more rigid, although it does have a little flexible hydrocarbon on the end. So what I'm going to be interested in is putting cholesterol into different bilayers formed of these lipids. Okay, now the cholesterol story. It's a very interesting, very complicated, very long cholesterol story, and you all know cholesterol is bad for you, et cetera, et cetera. The fact is that all your membranes have a huge amount of cholesterol, and if you didn't, you would not be healthy. So it's cholesterol in different places that really is important. Cholesterol really does exist. This is just, you can find pictures like this out of lots of different places. This is in Nature, which is supposed to be telling you where the fashionable things are, and it just tells you that cholesterol is a very active molecule in cellular biology. So I'm not going to take you through all those pathways and things. That's not what we do in a physics talk. What I want to talk about is the physical properties of bilayers and membranes, and with cholesterol. And the properties I'm going to list that I do in my experiments are the following. First of all is how thick is that membrane. I showed you roughly 4 nanometers, but it's going to be different for those different lipid molecules. And by the way, I forgot to mention this. One of the things that I find very appealing about this subject is that you have a lot of different small substitutions that I've emphasized on that slide with all the different lipid molecules, each one of which changes the properties a little bit, and that allows you, it gives you an experimentalist sandbox to play in, to do experiments to find out how the interactions are working between different small changes in the molecules. Now, all of those molecules are in your body. Okay, so the body is making use of all of those in some fashion. And the real question is, why do we need so many? Why wouldn't just one of them do the job? And these are questions that I'm not going to be able to answer, but they're the questions that are out there that we're trying to address. The way we're trying to address them is to look at the physical properties of these lipid bilayers. So the first one is the thickness, and I'll define that better a little bit later maybe. The next one is an order parameter of those hydrocarbon chains. And the order parameter traditionally in physics is the thermal average of the second order Legendre polynomial. Okay? So the beta here is the deviation from the perpendicular to the bilayer. So here's my bilayer. I've just drawn it as a slab. And here's the perpendicular. And the orientation, how far it deviates from the perpendicular. If you have a perfectly oriented bilayer, everything is straight up, you might say. But then as you get all those fluctuations, then you get deviations. And this gives you a measure of those deviations, which is called the orientational order parameter. Final one, which is, I think, even more fascinating than either of those two, is the bending modulus. So you have a bilayer which is going to start out flat, and that will probably be its ground state conformation, but it can bend. It's like a beam or a flat plate. It can bend. Of course, that costs you extra energy, but these energies are small compared to kT. And so there are going to be thermal fluctuations bending these membranes. And the question is, how big is the energy that you get? Well, the energy, the two-dimensional curvature is of course the two-dimensional Laplacian of the, okay, U is the deviation from flatness, and if you look at this is the curvature, and of course the energy can't depend upon whether it's curved this way or this way, so the lowest order energy contribution comes from the curvature squared, so that's this, and if you integrate that over the whole surface, there's still a phenomenological parameter, which is the Kc, which is going to depend upon the lipid molecules that you have in the system to begin with. Okay? So what we're measuring is this Kc for the energy of bending. Okay? And the question then is, how are these properties affected by cholesterol in those different lipid molecules? Okay, so here's where I needed the thunder. All right? Here's the dramatic result, and it's all on one slide, so let me take you through it. Remarkable results, okay? So that molecule that had saturated hydrocarbon chains is the one that most people, it gives the results that most people think of as being the appropriate results for lipid bilayers. So now what I'm plotting here is the bending modulus, how much energy it takes to bend the things, and by the way, here's 20 kT down here. And here's the cholesterol mole fraction. So if I start out here, I have the pure bilayer. Now let me get my notation. So this is the one I want to focus on. And so here's the pure bilayer. Its bending modulus is less than 20 kT. Here's 5 right here. As I add the cholesterol, it gets stiffer and stiffer and much stiffer, even at only 30% cholesterol. Body cholesterol contents go up to 50%, depending upon which membrane you're looking at. So that's the conventional behavior that everybody thought about. But we discovered by accident, not something I was looking for, we discovered by accident that when we looked at this molecule, which had one unsaturated double bond per chain, and now let me find that one, it's the round circles, the bending modulus, if anything, it might have even decreased, but within our error bars, it's essentially not changing. And that blew our minds, frankly, and as well as a lot of other people. Why did that happen? By the way, this is my graduate student who is the first author on these papers. And let's see, what else do I have here? Well, I'm just reminding you what the molecules look like here in case you've forgotten it already. Oh yeah, the second point is, if I go to a longer lipid with 22 carbons, it's also, and it also has unsaturated double bonds, it also is staying constant. But if I look at one that's in between, now let's see, SOPC, I didn't tell you that one before. It has 18, 18.0, 18.1 for the chains, and it is increasing as I add cholesterol, but not as much as the DMPC, which really increases a lot, but certainly more than the ones with both chains unsaturated. So the conclusion on this is that the number of saturated chains, or the number of unsaturated chains if you prefer, number of saturated chains is important in what happens to Kc when you add cholesterol. And I might add, this was such a striking result, and probably why I got to be a phys rev letter, but there have been two groups since then who have confirmed that the OPC Kc modulus does not increase with cholesterol. So the other properties that I mentioned that we study are thickness. So here's the thickness. I haven't defined that well yet, but basically don't have to. And let's see, if I look at the MPC, it's increasing rapidly. The thickness increases rapidly, but then it kind of stops increasing. Couldn't go any further, but our technique doesn't help us up here. For the one with one saturated and one unsaturated, it's, well, let's see, let me focus on the two unsaturated chains. And in that case, it increases, but increases much more slowly and takes longer to come to a plateau for both of these. And this one, which has one unsaturated and one saturated, is in between. Okay, so as expected, and everybody's known this for a long time, cholesterol thickens the bilayers. And actually, we have a second method that also confirms that. This was a postdoc with us recently. And this is this order parameter for the orientation of the hydrocarbon chains. And in the case of the MPC, it increases rapidly and then it plateaus out, whereas the unsaturated lipids is a much more gradual thing. And in fact, these two methods really give the same measure. And so if you plot one measure against the other measure, there are fairly linear correspondences between the two. So both of them are agreeing with each other.
Can I ask a question?
Sure. Oh, absolutely. By the way, anybody should ask a question at any time. Okay? I really appreciate that.
So,
yes, where is the question? Yes.
So these cholesterol molecules, are they evenly spaced over these membranes, or do they interact with that one? You don't know.
Excellent question. I wish I knew the answer, and I'm proposing to simulators that they help us to find that answer. Okay, and I will come to a slide where I'm going to make a hypothesis, a conjecture about that question. Okay? So one of the things that we don't know from my techniques is what's the lateral distribution of the different molecules in the mixture. My technique does not help me to tell that, but that's something we really would like to know. Yeah, so at any point, okay, we have a small enough group. It's too bad we don't have a small lecture hall. But, you know, feel free to engage at any point. So my point here is, from these techniques that we have, it takes less cholesterol. I like to do a play on words. Usually we say when something comes up like this and then it saturates. Okay, but I'm using saturate for another thing, which has to do with the chemistry of the hydrocarbon chains. But here I'm putting both words in the same sentence. It takes less cholesterol to saturate the saturated lipids and more cholesterol to saturate the unsaturated lipids. Okay, now I mentioned that I didn't mention it. This is the first time. There are theoretical fits. So if you look real closely, you can see dotted red lines here, which are theoretical fits to those experimental curves. Now, the model that we're using there is a really primitive model from freshman chemistry. It's essentially a model that assumes that there's a reaction between the cholesterol and the lipid, and they get together and they form clusters, basically, which in freshman chemistry is usually called a new molecule. And it's so simple, as a physicist, I hate to even promote it. However, it has enough parameters that it fits the data very well. And it may actually lead to some insight. The insight is that with the saturated lipids, the amount of lipid that a one cholesterol can complex is larger. It interacts with more lipids and has a larger complex than when you have one cholesterol in an unsaturated lipid mixture. And that's why this saturation level, why it levels off quicker at lower concentrations for these lipids, which are both saturated, relative to these lipids which have unsaturated chains. Okay, so again the conclusion, number of saturated chains is important for these properties. Now, this next one concerns a little bit the question about the lateral distribution, but doesn't completely answer it. What's the area in the horizontal direction, in the in-plane direction? What's the area of the different molecules in these mixtures? And this is a subject that Ollie and I got into, as I just mentioned. But in the literature, people doing simulations were doing all sorts of complicated things, tessellations, trying to figure out, drawing boxes around the different molecules and trying to find out how much area they subtended. Tessellations or Voronoi tessellations was a favorite way. But there's a unique definition for the areas in mixtures of lipids. And this is something which we call partial molecular areas. And it comes from old physical chemistry, 100 years ago. And so this is something that Ollie and I did together, published in 2005. The definition is you take the total area and you take the derivative as you add one more molecule of the type that you're interested in. Okay? Holding all the other molecules, all the numbers for all the other molecules fixed. That's a definition. You might say, well, all right, so we know how to do partial derivatives, all right, but what does it do for you? Well, I'm not going to rederive all of the old physical chemistry, but if you do this, you actually, by using homogeneity theory in a simple way, you come up with the total area is equal to what it should be equal to, which is the area of each lipid, each type of lipid, the I, times the number of those lipids. It's a sum of those things. Okay, that's not so obvious if you do things like this up here, but it's a unique definition. It's a thermodynamic definition. Okay, so Ollie and I published using simulations 4 years ago, and as we were finishing this work up, I realized that in the last sentence of our paper, I had put in the comment that we really should do it with experiments if we could, and we can't. So this is going to show what happens experimentally. So what we do is we plot the area per total number of molecules, both lipids and cholesterol. We plot that, and those are these symbols over here. And we plot them versus the cholesterol concentration. And let me see. In order to get these derivatives, and there's some stuff, don't feel badly if you don't understand every step here, because I'm not going to explain every step. Go read our paper if you want to see every step. But I'm going to just show you a little bit about this. If I take a tangent to the curve, so I'm looking at this concentration right here, and if I take the tangent, which I've drawn by this magenta line, and I extrapolate it up to here, that's the partial molecular area of the lipid with no cholesterol. That's the partial molecular area of the lipid. And if I extrapolate it down to here, that's the partial molecular area of the cholesterol. And so let's see if it works. So I go up here, I go over here, and that is in fact what I've drawn. So the lipid, the area of the partial molecular area of the lipid is this curve here. And my formula that I just gave you gives me this point here. And the formula I gave you for the cholesterol gives you this point here. So this is the partial molecular area of the cholesterol. Now, something rather dramatic is happening. And that is when you look at the cholesterol, if you go down to small cholesterol concentrations, its partial molecular area is negative. That says that you put a cholesterol in, the total area is actually condensing. It's not putting something in and making it bigger. It's putting something in and making it smaller in this direction. Okay, now, how can anything which is intrinsically, you might say, is intrinsically a positive quantity, how can it be negative? How can something take up negative area? Okay, and the answer is that the cholesterol orders the hydrocarbon chains of the lipids next to it and makes those have smaller area. But when you do it using partial molecular areas, that goes into the cholesterol molecular area. So it's There's a well-known example in volumes. If you take sodium, or sodium chloride, and you stick it in the water, the sodium ion will actually, by electrostriction, will draw the water molecules closer to it, and that will actually reduce the volume. So negative partial molecular volumes are well documented. In this case, we have negative partial molecular areas, and the mechanism that I just gave was, and you saw that on the previous slides, where the order parameters gets bigger, which means that the cholesterol, that the lipid molecules are becoming more ordered, which means that they take up less area. Okay. This is, I just explained this, and it's called the condensing effect. And the only thing that I didn't explain, this is for Ollie, is how do I actually get these areas from the experiment. And what we have is very accurate volumes for both of these things. Again, using the partial molecular volume idea and the concentration. And we have a pretty good idea of the thickness. And so these are derived quantities to a certain extent, but it works. Okay. And the final slide on this is to compare the different lipid bilayers with the different, and so by this time you're probably getting tired of looking at all these things, but there's an interesting difference again, that with the saturated lipid, the condensing effect really comes in very dramatically. It goes strongly negative. Okay, this is the one I showed you before on the last slide. I didn't tell you which lipid it was, but it was this one. And it became slightly negative. It has one unsaturated chain. This has no unsaturated chains, and it becomes strongly negative, whereas the two that I have with one unsaturated chain, one unsaturated bond on each chain, doesn't go negative. There is a condensing effect, but it isn't as strong. So again, the same motif, the same theme. Condensing effect also depends on the number of saturated chains. Okay, so now the speculation slide. Actually, some of this is well-known speculation in the literature. In fact, most of it is. So the cholesterol looks like this, but if you took a picture of it, or you make a molecular model of it, this is not a very good one, I'm afraid. There's a smooth side over here, and on this side you've got these CH3 groups sticking out, forming kind of bumps. It's not very dramatic. You don't see it well here, but it's the picture. And the saturated chain, if you draw a molecular picture, if you make it straight, this is the ground state. If you make it straight, which has the smallest area per molecule, by the way, that can pack easily against a smooth face on the cholesterol. Whereas if you look at those double bonds, and I didn't tell you something, again, organic chemistry, those double bonds are a particular type called cis. There is another type called trans, and you're not supposed to eat those. Okay, so you don't want to eat trans fatty acids, but the cis fatty acid has this great big kink in it, and I'm speculating here that that will pack better against the rough face. At least it will pack better than these things will against the rough face. And so here's a new hypothesis, and people will probably shoot it down in a few years, but my hypothesis is that when we have unsaturated lipids, the smooth faces don't like. The smooth faces are happier with saturated chains and not so happy. Happy means slow free energy, of course. They're not so happy with the unsaturated chains. And so maybe in the unsaturated lipids, the cholesterol dimerizes with its smooth faces against each other, and then the cholesterol complex would be two cholesterol molecules rather than just one. And that fits. And I'm not going to go into this in great detail. That rationalizes the number of the P number in the lipid cholesterol complex. So the usual picture is this is 1 and this is a number P, but this could be 2, and this would then be, this would account for the ratio being only half as large for the unsaturated lipids than for the saturated lipids. So some of these things are in this letter, and they'll be the full paper. Okay, so that's kind of a show and tell of what we do with cholesterol and lipids. But as physicists, you should be very unhappy because I showed you what we do, but I didn't show you how we do it. And how we do it is really not that straightforward. It involves a lot of physics and a lot of analysis. And so what I'm going to do now is the materials and methods section of our work. How am I doing on time, by the way? Okay. But I believe that this is important. And usually when I gave talks, I used to do this first. Everybody would go to sleep. And then I would show a few results at the end. So I'm reversing the order just to keep you off balance. And so the first issue here is, what about samples? Okay. Well, one of the reasons I got into this as a theoretician is it was really easy to make up the samples in the old days. You buy some lipid that was synthesized by a good company that makes good lipids, and you put it in some water and you shake it up and you got a sample. And the kinds of samples that you got that way were something called multilamellar vesicles. So each of these things is a bilayer, and there are many of them in a thing that looks kind of like an onion, okay, with all these bilayers wrapped around. And the nice thing about it is it self-assembles, so anybody can make these things. But when you do diffraction on these things, in diffraction it's called a powder sample, and the scattering X-rays go isotropically in all the directions, and that loses information which we don't want to lose because it doesn't allow us to do some of the things I showed you. We can also, in fact, we also do, we've stopped doing these, but we do unilamellar vesicles, and they're actually rather small. They're only this 600 angstroms of diameter, so this is actually the correct aspect ratio, but they're very useful for some things. But our main technique, our main sample preparation is using oriented samples that look, schematically, they look like this. So I have 1, 2, 3, 4, 5, 6 bilayers. There is a distance between, there's a repeat unit on this, which we call D. Sorry, here it is, D spacing. The repeat unit is the order of 6 nanometers. Now I told you the thickness was only 4 nanometers. That means that there's water in between the bilayers. And these are prepared, we have a neat way of preparing it. I won't go into details. They're prepared by the person who really runs my lab, and you can see there might be some relationship by the name. So those are the kinds of samples that we deal with. Oh, the point is there are a lot of these. Okay, I only showed 6 here, but there's something like 1,600 of them in our typical samples. And the experimental geometry that we have for this, for doing X-ray scattering, is we have a beam coming in like this. We use the chess synchrotron for lot of our measurements. And the sample is here, it's stacked up. We rotate the sample, and then we see, if we were just looking at crystallography, what we would see was spots that would obey the Bragg equation, which, let's see, I didn't do this very well. Okay, so here's the Bragg equation. The order H, so this would be first order, second order, third order, fourth order, and that gives us the scattering angles are given by the equation if I know what D is. Of course, what we do is we measure the scattering angle and find out what D is. The Q values, and I hope everybody has seen Q values before, but the Q values are essentially, well, here's the definition. QZ is 4 pi over lambda sine theta. Okay, let's see. I guess that's all I want to do there. Okay. Now, what we really want for biology is a single bilayer. Okay, and what I've just shown you was all these stacks. So the advantage, of course, for diffraction is I get a lot more signal if I have a lot more bilayers. But am I going too far away from biology when I do that? So that's the question, and it's a criticism that one can make for the using diffraction on stacks of bilayers. So let's see, let's see, what do I do here? Typical self-assembled arrays in water, fully hydrated, have enough water that, okay, so if I do that simple way of preparing them, I get something like this, where now let's see, what I'm going to do here is, this is the first, okay, I need to do something else here. Electron density is like this, in this direction, and this is the distance along the normal to the bilayer. So the electron density has peaks here, and then there's another bilayer over here in a different color, and it has peaks, and there's a repeat, the D spacing. And oh, this is what I needed. This shows you where those peaks correspond to the different parts of the lipid. The head groups are the ones that are electron dense, and so the lipid is packed like this, and here's another lipid over here, packed like that. The major point of this slide is that there's a lot of water in between them. Okay? And if there's enough water in between them, the interactions between the bilayers are small compared to the interactions within the bilayers, so the inter-bilayer interactions between the bilayers are not going to perturb the structure very much. Okay? So that's the main theme there, and that happens when we have so-called fully hydrated bilayers where we put them in water. So let's say, I already mentioned this point. Oh, sorry, I haven't mentioned this point. For X-ray scattering, it's advantageous not to have the X-rays go through a lot of matter because they get absorbed. And so for looking at these samples, we need to be able to make these samples hydrated to make them biological, but we don't want to put them under a lot of water because that absorbs the X-rays. So we need to be able to hydrate those samples from the vapor. And now this is an interesting, maybe I shouldn't take the time on this, but it's an interesting technical issue. And it led to a hindrance, it led to a slowdown of the field for a long time. Okay, so what I'm showing here is that when I have fully hydrated, when the relative humidity is 1, fully hydrated samples, I get a D spacing like this for DMPC. But as I lower the humidity by a little bit to 98%, sorry, 99.8% relative humidity, I've lost a lot of that water between the bilayers. I've lost something like 6 angstroms. Okay? And when I go down to 99% relative humidity, I've lost over 10 angstroms. And now the bilayers are getting close enough together that they start to interact, and we don't want that because most of this membranes in cells are not close together. Occasionally they are, and that's an interesting issue regarding interactions between bilayers, but for most of them we don't want to do that, so we want to be up here. However, for many, many years, starting with Nobel Prize winner Wilkinson back in the 70s and going through a number of other excellent investigators, people were not able to fully hydrate their lipids from the vapor. They were down here someplace. And it only became clear in the last 10 years that that was an experimental artifact which just required an extremely good hydration chamber to be able to get the full hydration. So it was an experimental artifact, and we really want to be up here because that's the biological interest. So, and just to emphasize that point further, if you dry them to 95% relative humidity, which sounds like they're really humid. I mean, if you're outdoors in 95% relative humidity, you really feel the humidity. However, lipid bilayers don't. And then what you get is the bilayers are crowded together, pushing against each other, and that changes the structure. And it makes it very difficult if you're interested in adding things to the lipid bilayers like peptides because there's no water for them to go into. So there are good reasons not to do that. And we managed to avoid that problem by having a chamber that we've constructed. This is not high-tech science, but for a previous theoretician, I'm proud of this, which is why I'm showing it. So there's a chamber here and the sample's in the middle of it. And let's see, to remind you, okay, so the X-ray beam is coming in through here, it goes into the chamber, the sample's in the middle of it. I'm not going to show you that. Here's the sample chamber. My former PhD student constructed this. And the other thing that's absolutely been very, very helpful in this field is having two-dimensional detectors. This is an early detector from Saul Gruner's group at Chess, We use a different one now, but this was starting 10 years ago, this was very important for the kind of research that we're doing now. And incidentally, so show you that I'm not really an experimentalist, you see, we had to use a kludge in our experiment to prop things up. So this is a block of wood. Well, wood is very popular in Sweden, so having wood in the apparatus is not so bad. Okay, so traditional diffraction methods, and now here I need to orient you. This is the QZ direction, which is perpendicular to the bilayers. This is the other direction, in-plane direction, and if you do ordinary crystallography, you see orders appearing here. Forget these things, this was our substrate. You see orders, we actually could get up to 10 orders for this DMBC, but this was in a phase called the gel phase, which is not the biologically interesting phase. If you do all this, you can easily get the electron density by taking the square roots of these things. You have to do some phase, solve the phase problem. There are ways to do that. I'm not going to take you through it. But you get beautiful electron density profiles. People have been doing this for years. This happens to be one of ours. And it works fine. The problem, okay, so now what I want to remind you of, what I just showed you was data for down here, which is cold for this particular lipid. It's in an ordered phase which does not really exist biologically. The interesting phase, the biologically relevant one, is up here, which is like that MD profile that I showed you, where all the chains were doing very disordered types of things. And that's the phase we want to study. But the problem is that you don't get orders. Well, you actually get an order here and an order here. I'll show you what these are in a minute. But after a while, you certainly don't get up to 10, which would be way up here. Instead, you get a lot of diffuse scattering. And that really messes up the analysis that I just indicated before. The crystallographic type analysis does not work for these fully hydrated, biologically relevant fluid phases. And instead, you get a lot of diffuse scattering. And so my message is that, okay, there's more information in this diffuse scattering than there is in the peaks. But where does the diffuse scattering come from? And it comes from fluctuations or disorder. And that's what I want to emphasize next. So this shows one of our Monte Carlo simulations, but it wasn't an atomic level simulation. It was a simulation where we modeled each of these things as a plate that had bending moduli. I'll show you the theory that gives you this in a minute. But what we have is samples which are no longer perfect crystals with the planes stacked up with a regular D-spacing. They have much more disorder and fluctuations in them. And that's the kind of thing that gives rise to that diffuse scattering that I showed you in the previous slide. So let's see, I think that's all I need to show you here. Here's the theory. So this is the smectic liquid crystal theory, which has been around for 50 years. De Gennes' book and so forth have it. And so what's shown here is each bilayer has been condensed to a simple plane. And this shows, okay, so if it was the flat stack position, the ground state would have all the bilayers flat with these spacings shown here. But of course, there are fluctuations of two sorts. The first sort is that the bilayers can bend. I've emphasized that before. But they can also compress or dilate in this direction. The way the measure we put on it here is, so this is the deviation from the mean position, the use of n of r's. And I've already shown you this term here. This is the bending modulus energy. This is an energy that has to do with the compression modulus. So the difference between the deviation here and the deviation here gives you a term here, which comes in harmonically with a so-called B, or compression modulus. So that's the theory of the smectic liquid crystal theory that in fact gives rise to the diffuse scattering. Here's the KC and here's the B with the two phenomenological moduli. This is the one I focused on earlier. This theory was worked out in physics by a Canadian named Caillet, who was working in De Gennes' lab at the time, or De Gennes' group at the time. And he did very beautiful asymptotics, which gave critical phenomena. So those of you who know statistical mechanics, in critical phenomena, there are power law behaviors. And this model also has power law behavior in the correlation functions. So usually things decay exponentially, but in this model, you're essentially at a critical point because the correlation functions decay with power laws. And he did a beautiful job of that. However, to make this, to use this theory to analyze experimental data, you need to do numerics. And I have some very, very talented graduate students who have worked this out, and we've done the numerics. That should be 5 slides, just to emphasize how much hard work we did. But I'm sparing you that because I'm getting near the end of the time that I have. So this is to emphasize the data analysis in this new method. So what I'm showing you here, again, QZ is now in the horizontal direction. I should reorient these things someday. This is QR. And this is the diffuse scattering, which I've emphasized with my grayscale. You hardly see the peaks under here. There are some peaks. But this diffuse scattering that we're focusing on. And I'm going to tell you now that the intensity that we see is equal to the product of a so-called structure factor, which has to do with the theory of the fluctuations between these bilayers. And that structure factor is, if you know the Kc and B, if you know the moduli, then you can calculate the structure factor. Okay, and it looks like this. Okay. This factor is so-called, we call it the form factor. Different people use different names for these things, but this is the form factor. And it's the Fourier transform of the electron density profile. So in real space, we have an electron density profile. I've indicated those before. But this is the Fourier transform of it because scattering is done in Q space. And now what I'm going to do is to emphasize, okay, the question, the problem here is I have data which depend upon 2 factors. How on earth do I get either of those factors from something which is only the product? And the answer is I have direction, I have orientation to help me. This one depends upon both QZ and QR, but this one only depends upon QC. And so if I go in the QR direction, this one is a constant. So if I go in the QR direction, I can learn something about S of QR. So I'm going like this in these directions, and I can do it for many, many different QZ values. And I can fit the data there. So fitting along QR, simultaneously along all these QR slices, I can determine Kc and B. I've already shown you the Kc, won't show you the B today. And then I can determine the S of Q, which is this up here. And now, and that, of course, the Kc and the B lead to interactions between bilayers and the flexibility, so I've already done that. But now I also want to do structure. Oh, and by the way, the data are very nasty along here because I get specular reflectivity from the substrate, and here I get mosaic spread, so I avoid those regions and I focus on this region. And then, I'm missing something. Oh yes, and then after I get this, I take the original data, the original I's, and I divide by this, and now I have the F of QZ, and that gives me the Fourier transform of the electron density profile, which if I then inverse Fourier transform it, I get the electron density profile. So that's the point down here, and then the electron density profile with the F of Q's lead me to structure. Okay. Well, that involves a lot of theory, a lot of analysis. How well does it work? And this slide is to emphasize, and this is actually old data, which were still noisier, and it works even better with newer data. But the point is, it works awfully well. So the theory is good. And I shouldn't go any further on that. Okay, so now finally I show you the structural information that I get out of our data, which is the F of Q's, which are now, this is for the OPC lipid. And there are many, many data sets here for oriented samples. The ORI stand for oriented samples. But also we've added on here the unilamellar vesicle samples, which are those small unilamellar vesicles. they're good down here, but because they're unilamellar and the scattering is not concentrated, they tend to not give you, they don't, the signal to noise on these gets bad when you go to high Q. But I just showed you on the previous slide that down here in this region, I had to wipe out a lot of the data because of mosaic spread and very strong peaks and lots of nasty things going on. And so the oriented data start right about in here, and they go all the way out to here, which is roughly twice as far as people have been able to get with multilamellar vesicle samples, including ourselves. So we're very happy that we can get this far in Q space, even though data get kind of noisy out here. At least we know that there are already data out there. So the F of Q, again, remind you, is the Fourier transform. And so now we can get the electron density profile. This actually involves, and since I'm running short on time, this actually involves a certain amount of modeling. This modeling here provides us with a whole bunch of, a lot of interesting numbers like the area of the molecule and other thicknesses, and you can find a lot of results if you're interested in going to our website. We're adding neutron scattering. So this, oh sorry, this is a recent postdoc, Norbert Kacherka. His name got missing there. And he's now at Chalk River and does neutron scattering. We're adding neutron scattering, and we're trying to, he has come up with a way of analyzing both the X-ray scattering and the neutron scattering with the same model. It's quite a feat, I think, and that's going to be, it's already being done. But modeling, no matter how you do it, it always requires assumptions. And that leads me to the last topic that I want to emphasize. Oh, sorry. So we're doing lots of other things, more lipids, temperature dependence, interactions. Mentioned cholesterol already. Adding proteins. We get money from the NIH, and they like HIV. And so we had some HIV peptides. I won't show you that today. But when we start adding things to make it more biological, we're getting more complex systems, but we have the same amount of data. Basically, we've got what I've shown you, the F of Qs, the KCs. And if you start doing modeling, as you add more things to the model, you get underdetermined very rapidly in terms of being able to say what the data mean. You have too many parameters, and your models become underdetermined. And so this is my this is why I'm here, and why Ollie invited me, and why we've collaborated and will continue to collaborate. There will be, I want there to be lots of synergy with simulations, because with simulations, and now again I apologize, Ollie, this comes from Scott Feller this time, but simulations tell you so much more than we can ever get out of looking through a glass darkly with experiment. It tells you where all the atoms are. I've only focused here on some of the groups that I've emphasized, the CH2 groups and the phosphorus group and so forth. And this tells you where they all are. Okay? But the problem is, do you really trust the simulations? And even simulators are quite realistic about this. And so the synergy is we can give them data that they can tune the simulations to, and they can then hopefully tell us a lot more about what our experiments mean. Now this is actually a slide that, okay, so again, interacting with Rich Pastor, we came up with something a couple of years ago, which may or may not be the best thing. But our emphasis was that since there may be small errors in the potentials, and in particular regarding the area, that there could be competing interactions, this is my take on it. We could get poor structure in constant pressure simulations, but what we thought was, well, let's change the parameters, or in this case, let's change the lateral pressure, or let's change the area per molecule to get the best agreement with the experiment. In fact, when we changed the area from 67.5 to 72.4, the electron density profiles were much better at this one than they were at this one. And so we thought, well, gee, that really confirms that the area is this. And that was using the other kind of force fields that Ollie uses, using charm force fields. But since we've added the neutron scattering, things have, sorry, I should get through this slide quickly. Things have become rather complex. And it's clear that the charm potentials are no longer working so well. So now Sagar Pandit, who uses the Gromax, the force fields that Ollie helped develop some years ago, has a recent paper where he's also getting good agreement with the electron density profiles, but with a much different area per molecule, which is actually agreeing better with our recent neutron results. So at this point, the talk descends into chaos. Okay, I don't have any bottom line for you, because this is really active research, and a lot of different groups and a lot of different experimental groups are very much involved in this question of trying to have the synergy between experiment and simulation work. So this is my most recent group. You've seen a lot of these people's pictures before. Let's see, this is my current graduate student. She just left a few months ago, and this is my Dr. Tristan Nagle. And this is the previous group, which had Norbert Kucherka and a very bright graduate student who did a huge amount of this development, this physics development that we needed. So I think that's, okay, I've gone over time, so that's a good time to stop.
Questions?
Yes?
Yeah.
Oh, sorry.
Can you say a couple of words about the Monte Carlo simulations that you skipped over?
Okay, they were basically like that theory equation. They were the mesoscale. So what we put in was a bending modulus. We also actually went a little further than that. Instead of having just the harmonic interactions between the bilayers, we actually put in what's considered to be the proper van der Waals attractive interaction and the repulsive interaction from the so-called hydration force. And what we were testing on those actually was the issue of the fluctuation force, the Helfrich force. So there's an entropic interaction between between anything actually. That's because, let's say this differently. As the bilayers undulate, if they're real close together, they can run into each other. And that's not permitted because that would be a very large interaction. And that gives rise to an entropic interaction which pushes them apart. And so that's the Helfrich repulsive force. And we were getting at that by doing the Monte Carlo simulations. That's a whole other talk, but that's basically was the reason why we were doing this. I just used it here to show the kind of structure that you would have if you have all these interactions. Sorry, did that help to answer?
Yeah, you also mentioned the possibility of disorder.
Yeah, I mean that was the point. To show all the disorder that should be there if the smectic liquid crystal theory is correct, and then using the smectic liquid crystal theory, we can predict the disorder that we actually see in the diffuse scattering. So we see all this diffuse scattering disorder, and we believe we understand where that's coming from.
Yeah, I was curious, you showed us that the effect of cholesterol in membranes built up of saturated, unsaturated effects is very different. Is there a biological effect that is explained by that?
Okay.
Is there a relation?
Yeah, that's an excellent question. And we actually do have a brief paragraph in the newer paper that we've written on that. It's not in the PhysRev letter. The fact is, okay, so that slide that I really went over very briefly, cholesterol is involved in a lot of trafficking in cells. It's an important molecule for so many different reasons. On the other hand, if you had fully saturated hydrocarbon chains in your lipids, and you need the cholesterol for other biological functions, now the bilayer becomes stiff. But bilayers have to do things. Okay, they have to fuse. They have to undergo different conformation, they have to be flexible. The cells do not like to be cast in concrete. A living cell likes to be flexible. Okay? And if you only had saturated hydrocarbon chains and you wanted to also have cholesterol for other reasons, you now have gotten yourself into a problem that the bilayers are too stiff. So my speculation is that the reason why we have unsaturated lipids in our membranes is because we want the cholesterol, but we don't want them to be too stiff. And thank you for asking that question. I really should incorporate that in my talk.
I just wondered, in this water phase, what pH and ionic strength?
Okay. Being a physicist, I like no ions, and we hope that its pH is close to, it actually is close to 7 for the neutral lipids. Okay, actually I should say, so I showed you that the lipids are dipolar, which means they're neutral on the whole. We don't need counter ions, and when you put them into water, they essentially stay at neutral pH. They're zwitterionic. But we are interested actually in charged lipids, and there are many charged lipids in cells. And of course then we have counter ions, and pH goes out of all over the place unless you put in some salt or put in some buffer. Okay, I've had a postdoc who always put buffer into everything, but I personally like to keep it in pure water. So everything you've seen here is in pure water.
Okay, thank you.
Thank you for coming.
And if there is anyone that has more questions, Professor Nagle is available here tomorrow. Please come to corridor A4 or contact me.